This demo presents an AI-powered workflow. It supports biomedical research and GWAS analysis. We begin with gene search. The researcher enters a biological question in natural language, without constructing a complex database query. The AI research agent interprets the intent, searches relevant evidence, and organizes the response around genes, molecular functions, and supporting literature. Rather than returning only a list of papers, it produces a structured insight report. The report highlights the main findings and their biological context. The report can be reviewed section by section, while source links remain available for verification and deeper reading. This shortens the path from an initial question to a traceable, evidence-based summary. Knowledge extraction converts the retrieved evidence into a connected representation of genes, concepts, and publications. The graph makes relationships easier to inspect, filter, and compare, while keeping each connection linked to its evidence. Researchers can move from a graph node to its source paper or report. This preserves context throughout exploration. Literature dialogue provides a focused way to work with individual documents. A paper can be opened and questioned directly. Answers are generated from the document context, helping users locate mechanisms, findings, and supporting passages without manually scanning every page. For broader questions, the literature review workflow starts from a research topic and expands it into a systematic search. The agent identifies relevant studies, synthesizes recurring themes, and organizes the material into a review with a table of contents. The generated report includes an abstract, thematic sections, methodological context, and references that can be inspected independently. This provides a reproducible starting point for expert revision, rather than treating generated text as a substitute for scientific judgment. Evidence management keeps intermediate artifacts in one workspace, including papers, extracted relationships, figures, and structured results. The system can extract gene relations from an image or obtain gene information from a full-text PDF for later reuse. The second half of the demo moves from literature intelligence to GWAS analysis. The researcher describes the analytical objective in natural language. The agent translates that objective into a sequence of tasks, requests the required data, and exposes each step of the workflow. Uploaded files can be selected and checked before analysis, reducing ambiguity about which genotype, phenotype, or intermediate result is being used. Intermediate figures and result files can be opened directly, allowing the researcher to inspect diagnostic plots before continuing. The workflow then proceeds through variant annotation, candidate gene mapping, and the generation of analysis-ready outputs. At each stage, the conversation records what has completed, what data were produced, and which action should follow. This makes the pipeline easier to audit and rerun. Researchers can still intervene when parameters need adjustment. The same applies to biological assumptions. The final outputs combine candidate genes, workflow status, and visual results, connecting statistical analysis back to interpretable biological evidence. Together, these capabilities create an end-to-end research environment, from literature discovery and evidence extraction to functional insight from GWAS data. Proof Gene connects literature evidence with functional insight from GWAS data. Thank you for watching.